Hey everyone, plasminogen activator inhibitor 1, or PAI-1, is a driver of aging and many age-related diseases, but it's a biomarker that not many people know about. To help change that, today's guest is Dr. Doug Vaughan, who has studied PAI-1, or PAI-1, for almost 40 years. Dr. Vaughan is the director of the Human Longevity Lab and is a professor emeritus at Northwestern University. So welcome, Doug, and let's get into the presentation.
Thank you so much. Exciting to be here with you today and to talk about my favorite topic. So I got into the field of aging sort of naturally. I came to it through a long journey and progression in my own science and my own learnings, really starting in the late 1980s. But let's start with a couple bits of background so we're all working at the same level here and we can think about this. Age. It's something we all deal with eventually. Sooner or later, you look in the mirror and you notice that you're aging. But there are two different versions of aging. There's a chronological age and a biological age. So this little video here depicts what drives chronological aging, right? Every time you complete an orbit of the sun and you have another birthday, you add another year to your chronological age. There's nothing you can do about it. It's cosmically indifferent. It's going to happen whether you like it or not. And as we're spinning and orbiting around the sun at 100,000 kilometers an hour, this is one part of the process that drives our aging and our measurement of biological age. But beneath that, and beneath the chronological aging, there's another clock that we're learning to measure and quantitate, and that's your biological age. That's the rhythm of where our planet's orbit lies. Beneath that rhythm of our planet's orbit lies another clock. It's biological, it's mutable, and it's uniquely our own. It doesn't count years, it counts change. So I hope everybody that's listening now has some appreciation for the difference between chronological age and biological age. And everybody can be plotted on one of these trajectories that I'm showing on this slide, where we look at biological age versus chronological age. You think about people in your family, people that you've known in your life, people that you've worked with. You might know somebody that's a super ager, somebody whose biological age seems to be much less than their chronological age, and they go on and on forever. We all want to be there. They're able to do what they want to do into their 90s. They're cognitively intact. They're still playing golf. They really don't need our help too much. Most of us fall somewhere in the middle where our biological age and our chronological age sort of match up. And that's sort of the typical pattern. And then there are people at the other end of the spectrum, the purple arrow here, where their biological age exceeds their chronological age. So they've got challenges around that. And this is really important because age is the most important risk factor for every disease we deal with in adult human beings. But the version of age that's most telling is your biological age. And the idea illustrated on this slide is that aging doesn't have to be the same in everybody. Makes me think, and I think probably makes most of your listeners think that maybe we can do something about it. And that's kind of the world we're living in today. So we're at a unique moment in human history. I was flying back from Greenland, from the UK a couple years ago, flying over the southern tip of Greenland before I knew it was going to be so interesting in the news as it is this year. But what I saw at my window were two gigantic glaciers converging and going out to sea. So I want to make a point about convergence. On one hand, the biology of aging has largely been demystified. It's not complete, but we know a lot about what drives aging in humans and in other animal species. Simultaneously, we have the ability to measure biological age with a precision that never existed before. And when those two things meet, as these glaciers do in southern Greenland, it makes us think we can do something about it. And that's the world we're living in now, and that's the science we're involved with as well. So this is the key idea. This is a geroscience hypothesis in a single figure. The idea that if we can slow down the process of aging, we can extend the health span. We can provide more disease-free years for everybody. And that's what our institute and our program is focused on. We're not necessarily trying to find a way to get people to live to be 130. We want to find a way for people to live to be 85 without having age-related morbidities. So I'm going to tell you a little bit of a personal story about how I got involved with aging. And, it's a little bit circuitous, so bear with me as I show you a few slides that sort of tell the story. So I'm a cardiologist. When I was doing my training in Boston in the middle part of the 1980s, the the um, the topic that captured everybody's imagination was the development of clot dissolving drugs like TPA. And with those in hand, we could do things acutely for things like an acute myocardial infarction, a heart attack, or a stroke. We could deliver these drugs intravenously. They could catalyze the clearance and the breakdown of those clots, and they could restore blood flow to those organs. So myself, like many cardiologists, was fascinated by this system and the impact it was making on my specialty. The laboratory that discovered TPA and actually had the patent for it was in Leuven, Belgium. And it was really pioneered and identified by this guy I have an arrow on right now, Desiree Collin. And so, I was so enamored with this concept. After I finished my cardiology training, I got a grant from the American Heart Association to go work in Desiree's lab for a while. And because I wanted to be at the epicenter of thrombolysis. And I went over there with a few ideas in hand and certainly a willingness to learn and great expectations about what an experience like that might be like. Pretty soon after I arrived in Belgium, I was having a conversation with Desiree in the hallway. He asked me what I wanted to do while I was there, and I gave him an answer. Uh, he wasn't very impressed. Um, and so he sort of stood there and looked at me for a minute, and he said, recently this rapid acting inhibitor of TPA has been identified and described, PAI-1, the protein you mentioned as he opened the show today. He goes, he said, he pointed over the freezer. He said that we have several milligrams of recombinant PAI-1 in that freezer over there. He goes, nobody knows what it does. Why don't you work on that? And I stood there for about 3 or 4 seconds, and I said, okay. That was 1988. Little did I know that almost 40 years later, I'd still be working on it. So down in the lower right-hand panel is a ribbon structure of PAI-1. It's a protein that circulates in your blood. Everybody has it. It's made in a variety of different places in our system, in the liver. It's made in fat. It's made in the vasculature when it's injured. It's made in some other places as well. It's got a short half-life, and it's cleared in the liver. The most important determinant of your plasma PAI-1 level is your body mass index, interestingly enough. So if you just think about the world today versus 100 years ago, there's a lot more PAI-1 walking around on the street in America and other places because of the increase in obesity and diabetes that are associated with increases in PAI-1 levels in the blood. So I devoted myself to working on PAI-1, and when I came back from Belgium and set up a lab in Boston, and then I moved to Vanderbilt in 1993. I was really dedicated trying to understand its role in cardiovascular disease and clotting. What my frame of reference wasn't very broad, and my bandwidth wasn't very extended at that time. But a couple reports appeared in Science over the next few years that sort of changed my orientation and opened my mind to possibilities around the idea that PAI-1 might be involved in something other than controlling the system that dissolves blood clots in our body. And at first was a paper from in the New England Journal, a small report came out around the holidays in 1992. Dave Ginsburg was the senior author, but key collaborators were Amy Shapiro and Bill Fay. And they described a young woman in an old order Amish community in Adams County, Indiana, who had an odd bleeding disorder. She wasn't hemophiliac in the normal sense. She didn't bleed spontaneously. But if she was injured and formed a clot, her primary hemostasis worked just fine. She would stop bleeding. But a day or two later, she would start oozing again and start bleeding, and it was hard to stop. And when they worked her up, they thought, well, maybe she's got a fibrinogen disorder or she's got a platelet disorder. None of that was true. Those were all fine. What she ended up having was no PAI-1, none. And she had a brother with a similar abnormality and a cousin with a similar abnormality. And it turns out there were several people, at least a dozen people in the larger extended Amish community that had this strange disorder related to complete PAI-1 deficiency. Now, I read that paper and I said, you know, I work on PAI-1. That's what I'm making my research career about this. Some way, somehow, I got to go study those people. They're going to teach me something about this that I don't know, but I didn't know what the question was I wanted to ask. But I filed that away in an important place in my mind, and I didn't forget about it because I said someday I want to go back and study them. But I had to find the right question. And the question that there was another report not long after that report in the New England Journal that helped change the question or helped frame the question for me. And it was a report from a Japanese group who were tinkering with the mouse genome and trying to insert a new gene into their genome that affected ion transport. Well, the transgene that they put in the mouse happened to land in a uncharted territory in the mouse genome. And when we looked at the mice that were generated by uh, this engineering, they had a weird phenotype. It looked like aging. The transgene had landed in the upstream regulatory region of a gene that had never been identified or described. You know, the genome of mice and humans was all being assembled then in the 90s, but these mice had a short lifespan. They only lived a few months rather than 2 or 3 years. They had growth retardation, skin and muscle atrophy, neurodegeneration, vascular calcification, hearing loss, all kinds. They looked like they were progeroid. They aged rapidly. And soon after this original report came out, came a secondary report saying that these mice, interestingly enough, have elevated levels of PAI-1 in their blood and in their tissue. So I took that information and I said, well, I wonder if PAI-1 is contributing to that aging-related phenotype. Does it contribute one way or another? That didn't come out of thin air. There had been reports published in the mid 1990s and from several groups around the world that PAI-1 was one of the key parts of the molecular signature of senescence. When cells become senescent, they lose their ability to proliferate. They lose their ability to repair and regenerate. And senescence is thought to be one of the key drivers of aging and aging-related morbidities, right? It's one of the molecular hallmarks of aging. So I already knew that. I said, well, okay, PAI-1 is involved in senescence. It's increased in these animals that have an aging-like phenotype. What would happen if we took PAI-1 out of the equation? So actually, after I moved to Chicago in 2008, we had the resources and the wherewithal to be able to do a complicated uh, study crossing different lines of mice. And we generated clotho deficient mice. Those are the mice with the rapid aging phenotype. We crossed them with mice that either had partial or complete PAI-1 deficiency. And we were to generate multiple lines that had clotho deficiency and either complete or partial PAI-1 deficiency. And we did a variety of things to look at them and see what happened. And it turns out taking PAI-1 out of the equation, even taking one copy of PAI-1 out of the equation, extended the lifespan of the clotho deficient animals, which we see in the orange graph here. It almost quadrupled their median lifespan, almost quadrupled it. And together with that, it also protected them from many of the aging-related phenotypic changes that you that you normally see in clotho deficient mice. So this was really, really surprising. It this was kind of a thunder, a lightning strike, certainly. And this is a painting by Magritte, but this could have been a selfie of me after I saw that data because it exploded in my mind. And the idea the idea that PAI-1 was a critical driver of aging and aging-related pathologies went off in my mind. And you probably can figure out where this is leading. Remember that other story I told you about, that paper I read in 1992 about an extended human kindred in Adams County, Indiana, that had PAI-1 deficiency. And then all of a sudden, I had I had a clear question in my mind that I wanted to ask in that Amish community. So members of an Amish community immigrated to northeastern Indiana in the early half of the 19th century. They came from in and around Bern, Switzerland. So they speak a Swiss German dialect with each other. They're very traditional in their culture and habits, the way they dress, the way they transport themselves with horse and buggy. They don't use combustion engines. They don't use electricity. But they are geographically and culturally isolated from the rest of the world in a variety of different ways. And the community in 2026 is comprised of about 20,000 people. It's only about a 3.5 hour drive from Chicago, amazingly enough. Now, just this is sort of a joke, but I don't know if there are other communities in the world that might have a PAI-1 deficient gene in them, but I don't know if they're available to me or if they would be reachable. If it was in Timbuktu or who knows where, we couldn't have studied them. But this is relatively convenient, and we can get there by driving in a relatively short period of time. It turns out that a substantial number of people in this community have at least partial PAI-1 deficiency. Everybody that carries the gene can trace their ancestry back to a single guy. He married into the community in the middle part of the 19th century. Listen to this. He had 17 children and 142 grandchildren with two different wives. And you fast forward 6 generations, you can figure out that there are probably at least 3,000 carriers of this genetic variant. And but they don't know it. The carriers, and this is really important, don't have the bleeding abnormality. They're completely unaware of the fact that they have this genetic variant in their system. Um, the the genetic variant is caused by a simple typographical error in their DNA that was provided by that original ancestor. So it's a duplication of two bases, A and T, in their gene, and you can see it here on the right-hand panel. Look under this A column, you see the three A bands here. There are only two in a normal PAI-1 gene. Same goes with the T, 3 and 2. Just inserting those two bases um, changes the fundamental configuration of the DNA, and it shifts it off code, and it creates a premature stop codon downstream just a little bit away. So if you carry one of these copies of this mutant allele, that allele doesn't make any PAI-1. Now, if you have one normal allele and one variant allele, you have half the normal amount of PAI-1 in your blood. If you have two of the variant alleles, you have no PAI-1 in your blood. So all the people that carry what the PAI-1 this PAI-1 variant can trace their ancestry back to that single guy. Now, this is an amazing story, and the Indiana Swiss Amish are a a remarkable example of a founder population. Well, what do I mean by that? Well, a genetic variant is overrepresented because the variant was entered in the community when the community was small. So about 10% of the Indiana Swiss Amish carry at least one copy of the genetic variant. If I was to go to Austin, Texas, or to Chicago and genotype people looking to see if similar genetic variants exist in the world at large, I would have to genotype about 100,000 people to find one. 100,000 people. But 10% of the Amish community in Adams County carry it. So 10% of 20,000, so probably at least 2,000, there may be more. So it's a 10,000-fold enrichment. So you know what? In a situation like that, Mother Nature has the opportunity to test a hypothesis, right? Does this genetic variant have an impact on their health and survival over time? It is Mother Nature tinkering in a way that happens to provide potentially a positive effect, not a negative effect. Most of the genetic variants we find in the world are deleterious, and they're found because there's a problem associated with it. And of course, the original proband that was identified with this genetic variant had a problem. She had a bleeding disorder, but all the other people that are carriers don't have that problem. So after after we completed that work with the mice and knowing that PAI-1 could protect them from aging, we applied to the National Heart, Lung, Blood Institute. We were able to secure a grant to actually go over and study the Indiana community. And together with Amy Shapiro and her colleagues in Indiana, who introduced us to the community, on a couple fateful days in early May 2015, we took a large group of collaborators and investigators and scientists and equipment from Chicago and drove over to Adams County and set up a temporary laboratory in a community center. And over 2 days, we studied 177 members of the community, amazingly. I mean, that included drawing blood, doing echocardiograms, measuring blood pressure, doing pulmonary function tests, all kinds of things. You can't imagine how complicated that was logistically to get that done. But we did, and it ended up being, you know, more interesting and exciting than I ever actually imagined. So we ended up publishing a paper called, and the title was a null mutation in SERPIN that's the gene that codes for PAI-1, protects against biological aging in humans. And what we found that day, and what we've found in continuing studies, we operate a satellite laboratory over in Bern, even to this day. What do we find? We found that the carriers of this genetic variant have longer telomeres. They're about 10% longer than their unaffected kindred in the community. They have preservation of their juvenile epigenetic patterns. They have lower fasting insulin levels. If you were to ask, what pathway would you want to hit and to potentially have an effect on biological aging in humans? I think a lot of experts in the field say, well, probably need to do something around insulin. Well, it turns out they have lower fasting insulin levels. They have lower hemoglobin A1C levels, so that is reflective of their more efficient metabolism. They're very protected from diabetes. The unaffected or the non-carriers in the Amish community uh, have a prevalence of diabetes of about between 8 and 10%, which is pretty normal for a Midwestern human population. So far, studying nearly 100 heterozygous carriers, we found one with diabetes. So it's a highly significant impact on the development of diabetes. They have preserved cardiovascular function, that is, their vascular system doesn't age as rapidly. Interestingly enough, they have lower PCSK9 levels. We didn't have any idea that PAI-1 was involved in the regulation of PCSK9. We know that now. And most importantly, they have a longer lifespan. They live about a decade longer than their unaffected kindred. And the whole concept is sort of illustrated for you in this little graphic right in the center of the screen. And it's a bell-shaped curve, and it sort of makes the point that there's an optimal range for PAI-1 for all of us. And let's set it up. So as I mentioned a little while ago, a lot of people in the world have too much PAI-1. And PAI-1 is an important contributor to cardiovascular aging, to clotting, to metabolic disorders, to cognitive decline, emphysema. It's even an important risk factor in several different kinds of cancer, like gastric cancer, pancreatic cancer, ovarian cancer, triple negative breast cancer. So too much PAI-1 is bad. At the other end of the spectrum, having no PAI-1 is not good either. You have that bleeding disorder that we talked about. Turns out people with no PAI-1 at all develop spontaneous scarring in their heart. And we've seen that in mice and in humans. It also impacts upon innate immunity. So you want to have your PAI-1 lowered, but not too low. And that optimal place brings you to a zone where your organismal fitness is enhanced. And that's kind of unique. I don't know that there are any other genetic variants that are well described in the world to provide this kind of protection and potentially long-term impact on health and longevity. We've gone on to understand that PAI-1 is involved with a lot of different systems, and that's part of what makes it interesting. If you talk about the work that's being done in the space, trying to discover novel pathways that might be targeted to impact on human aging, people always say, well, we want to find factors that hit on multiple systems, that hit on metabolism, that hit on senescence, that hit on mitochondrial function, do all this, and hopefully, and the work to find those sort of things and the intracellular level is going on and at the nuclear level as well. But none of those things are anywhere close to becoming testable in humans, I think, anytime soon. But it turns out PAI-1, by virtue of the fact that it inhibits or that it regulates the post-translational proteolytic processing of a lot of different peptides that are in our system, it impacts a lot of different biology. So, for example, IGF-BP3, top of the list here. It's an important driver of senescence in cells. Well, how do we normally get rid of IGF-BP3? Well, it's actually clipped. It's actually degraded by plasminogen activators, and plasminogen activators are inhibited by PAI-1. So PAI-1 enhances an IGF-BP3 signal. It enhances senescence. Um, Another one that's really interesting with regard to aging is BDNF, a peptide in your brain. You have to process that. It's generated as an inactive peptide. It's actually clipped by TPA. And when it's clipped, it allows you to make a new memory. It's important. It's very important in neuroplasticity. PAI-1 blocks that clipping. If you have too much PAI-1 in your brain, you can't make a new memory. PAI-1 is involved in inhibiting the activation of a very important peptide called HGF that regulates cellular proliferation. I told you a little bit ago, by surprise, we found that PAI-1 regulates the degradation of PCSK9. And if you look in the Amish population, the unaffected individuals in the community have a relatively high PCSK9 levels. The heterozygous carriers have a lower PCSK9 level. And actually, the homozygous individuals with no PAI-1 have the lowest PCSK9 levels. So PCSK9 is a huge and important target in preventing cardiovascular disease. And it turns out that PAI-1 regulates it. PAI-1 also regulates other peptides that are involved in lipid metabolism. So just look at this list. They say, oh my God, here's this very interesting peptide or protein. We know it's involved in regulating the clot dissolving system, but it's involved in other systems that are really important in aging. And I think frankly, Michael, I think we've just really begun to scratch the surface in understanding all the different pathways that might be impacted upon PAI-1, but it sort of starts to come to make sense. Say, oh, this is how it happens that lowering PAI-1 has such a beneficial effect in humans on aging and aging-related changes. I won't spend a lot of time on this, but just to highlight the fact that there are cell autonomous and non-autonomous effects of PAI-1 just on senescence. PAI-1 regulates the nuclear stabilization of p53, which is a key driver of senescence. It regulates the nuclear retention of cyclin D1, which impairs cellular proliferation and growth. Outside of the cell, we've already talked about IGF-BP3. A very interesting finding recently has been described by a few different groups around the world that PAI-1 increases the expression of PDL1. This is an important factor in immune checkpoint activation. And by increasing PAI-1 production and increasing PDL1, you lose your ability for immune clearance of senescent cells. Now, that's a very interesting component of the PAI-1 story and probably is a key driver of the role of PAI-1 in senescence and tissue aging. So we've taken what we've learned in Indiana. We've brought it back to the lab here in Chicago. We've done additional studies recently. This is from a paper we published in the Journal of Clinical Investigation just in December of this last year. We had some more information from our human the humans in the Amish community. We know now that they have uh, a reduced pulse wave velocity in the aorta. You see that right here in the upper left-hand panel. That means their aorta retains its flexibility as they age. And pulse wave velocity is probably the most important marker of vascular stiffness and vascular aging. So they're protected from vascular aging in a variety of ways. Now, we use CRISPR to engineer a new line of mice that have a genetic variant exactly like we see in human beings, exactly like we see there. And what do we see in them? Well, if they have one copy of that genetic variant, their plasma PAI-1 level is reduced by half. The pulse wave velocity in these animals is reduced as they age, so it protects them from vascular aging. And look at this. If you take away one copy of the PAI-1 gene using this CRISPR technology, you actually extend the lifespan of the mice by almost 20%. So it perfectly replicates what we learned in human beings but a reverse engineering tool has helped us to understand this even more. There are other ways that other people have looked at PAI-1 and its relationship to aging. This comes from a really classic paper now from Steve Horvath's group published in Aging in 2019. This is the story about grim age and that epigenetic clock that is probably the gold standard for measuring biological age in 2026. One of the key components of grim age is a component called DNA MPI-1. It's an epigenetic estimator of your plasma PI-1 level. There are several other factors that contribute to grim age, but DNA MPI-1 alone was the best predictor of overall mortality, the best predictor of time to coronary disease, the best predictor, look at these p-values, of comorbidities, and it predicted the development of high blood pressure and diabetes, heart failure or coronary disease, age and menopause, visceral adiposity, fatty liver. Now, I called up Horvath after this paper came out, and I said, this is fascinating. You have predicted with this mathematical model and epigenetic estimators exactly what we've learned in biological experiments. And Steve was kind of candid about it. He goes, I really don't know too much about what PAI-1 is. We solved this problem mathematically by looking at epigenetics and picking, you know, picking some rational markers that we thought might be involved, but I had no idea it would be involved in processes like this. I said, well, maybe we come from opposite ends of the pole of the story, but we end up in the same place. So Mother Nature's doing something very interesting in Adams County, Indiana. I think it might be an example of human evolution in the 21st century. If you think about it, it meets all the criteria for something that might be a heritable variable that provides an evolutionary advantage. It's inherited in a Mendelian fashion. There's a phenotypic consequence of reducing PAI-1. It has immeasurable effects on telomeres, insulin sensitivity, cardiovascular function, extended lifespan, all the kind of things you'd want to have in the 21st century. There's a potential fitness benefit over the long term. It's going to take a long time to know if this really has an evolutionary advantage. It may take millions of years. But what we're witnessing is a naturally occurring longevity mutation that occurs real physiological benefit. we don't know its evolutionary trajectory yet, but it's fun to think about. Fun to think about, and to the idea that, you know, human evolution may not be complete, but it's only going to happen in places that are where there is the opportunity for it, including cultural and geographic isolation and the ability to enrich a particular genotype. So it was that background, it's that story, it's that sort of dedication to this science that led me to where we are today here in Chicago at Northwestern. Our faculty, um, had developed a consensus several years ago that one of the topics we wanted to be good at, one of the things we wanted to invest in and develop was healthy aging. And our dean, Eric Nielsen, and others decided to create a longevity institute here. Uh, we received a generous gift from John Podestak and his family to create this institute, and it's comprised of several different centers that touch on aging and longevity. We have a Claude Pepper Center here in Chicago. It's one of, over just a dozen of them in the country. We created a Center for Population Science and Aging, Center for Nanoscience and Aging. Mr. Podestak is particularly interested in the relationship between HIV and aging. HIV, people with chronic HIV infections age more rapidly. He wants us to figure out how to slow that down. And we've extended that in a larger sense to thinking about doing or finding ways to slow down aging in people that are disadvantaged because of chronic conditions or circumstances. And I think that differentiates our program and our philosophy perhaps from others around the world. We do basic science and aging. And then we have this thing we call the Human Longevity Lab. And I think your listeners today will be interested in this. Now, earlier I mentioned that we have the skills, we have the tools to be able to measure biological age in 2026 that didn't exist before. Now, if you wanted to get your biological age measured just a few years ago, you had to go to somebody like this. Maybe they could read your palm, they could tell you where you're headed, tell you how long you were going to live, maybe tell you other things about you. But the science behind this is a little bit mysterious. Uh, but we have better ways today. We've created a human longevity laboratory here that's up and running, and it's really patterned after what we learned to measure and the things we learned to do in Indiana, in Bern County, or in Adams County. So what we do is I have multiple colleagues that are involved with this, and I was chair of medicine here for 15 years. When I stepped away from that 3 years ago and was asked to take on leadership of the Longevity Institute, one of the things I wanted to have was this version 2.0 of a longevity laboratory. And we bring our learnings as physicians to this, as well as the latest technologies and clocks. So we do functional and physiological testing on every system that we can get to relatively non-invasively. We couple that with deep molecular profiling, including various versions of epigenetic age. We've just acquired the technology to do broad platform proteomics with the O-Link system, and we're just getting that up and running. We also have the ability to measure something here called glycan age, the glycation of your immunoglobulins. It's another version of biological age. And then we also have a variety of different AI-based clocks that we use that measure things like take pictures or measure your biological age with photographs of your retina or your electrocardiogram. And with all that information, then we can talk to someone and tell them which one of these trajectories they're on. Are they looking like they're going to be around for a long time and pretty healthy? Are they on this? Are they more like this in terms of normal aging? Or are they an accelerated age? And as I mentioned, we're really focused on those individuals. This is so much fun, and I want your audience to see about this. So we utilize 4 different AI-based clocks in the laboratory. We do one that involves an analysis of 12-lead electrocardiogram. We work with a group in Brazil led by Tom Ribeiro. They've analyzed millions of electrocardiograms from humans now. And using machine learning, their tool can analyze an electrocardiogram and tell you your cardiovascular age or your biological age. Now, I'm a cardiologist. I can read an electrocardiogram. I can't tell your age, but this cool tool can, and we can put that up in the cloud, and we can get your age back in a matter of seconds. Do the same thing with retinal imaging. Simple camera. You look in an eyepiece. We snap pictures of your retina. We work with a group in New Zealand called TokuEye. They've analyzed tens of thousands of human retinal images with machine learning. Again, same idea. We put the images up in the cloud. Almost immediately, we can get back your biological age based on retinal images. It even has approval as a tool that can predict kidney disease. We work with the Oxford group at Ultramics in echocardiography. We do a simple four-chamber view of the echocardiogram. It's really simple and it's done using AI guidance to get the right images. They can analyze this simple image with an echocardiographic device that only costs a few $1,000s rather than $100,000. And we can tell you your cardiac age or whether or not you have any evidence of aging related changes in your heart. And then the one on the far right here is one of the ones I most enjoy showing to people. My colleague James Cotton at Shirley Ryan Ability Lab here is a physician scientist interested in quantification of musculoskeletal function. And he's developed a tool using an iPhone, simply collecting a video of a participant walking 10 meters forward and backward, and we can get things like your get up and go time, your gait speed, other aspects of your musculoskeletal function, all with an iPhone, and it's like I don't think there's anything like it in the world. He's really brilliant, and we do that in on our testing. So um, the point I want to make here is that, and I think this is really important to think about, that there are a lot of opinions about what's the best way to measure biological age in people. To be honest with you, I don't know. Um, that's why we measured in a number of different dimensions. We do functional and physiological testing. I think that's key. The things that bother us the most as we age tend to be functional and physiological changes. Our gait slows down, our grip strength diminishes, our hearing goes bad, our eyes uh, develop problems, whether it's glaucoma or myopia or cataracts. And then, you know, but there are molecular tools that can tell us our biological age, and they're great. And we use a variety of different ones that are important. And I think they're certainly the gold standard today. The broad platform proteomic platform, the tools like Olink or SomaScan can tell us a whole different story. It can tell you your systemic age as well as the age of all your organs. Glycomics give us another readout on biological age, and it doesn't always match up with proteomics. And then these AI-based clocks give another one too. I think all of them, at least today, we think give us our best estimation of your integrated biological age. And if anybody tells you they have the perfect solution to measuring your biological age, I would challenge that. I don't think I know, but let's imagine, let's imagine you come up with a therapy that you think improves your biological age or even turns your clock back. And you do the testing because it shows that you turned your epigenetic clock back 10 years. Well, is that associated with any functional and physiological changes? Does it impact upon any of the proteomic changes that we see or organ-specific changes or on the AI-based clocks? I don't think if those don't change too, I don't think you got any younger. I mean, I belong into that group in this discussion, and this is a lively discussion going on around the world, and the answer is not solved yet. For the time being, we're keeping an open mind about this, and we think it's better to collect more data, to collect more information about what's going on in the space, and use that in a real way to figure out biological age in people. So where do we want to go with this? I think maybe somebody that's listening is that say, what are you going to do about it? Just measuring biological age, is that the end in itself? Well, no. We want to create a global network, a network of laboratories similar to ours in Chicago, with the same kind of tools and the same kind of expertise. And we want to create an intervention testing network like the one that exists for mice in the US for the last 30 years. I think it's time to have an intervention testing network in humans. And we've already lined up and have formal agreements with several places around the globe to get this network up and running relatively soon. We have our own program in Chicago, another program at University of Illinois in Chicago, just across town. We're working with leadership at Mount Sinai Medical Center and in Miami. We'll probably have something out on the West Coast relatively soon. We've got two different groups in South America, either in Brazil with Tom Ribeiro or the medical school in Montevideo, Uruguay. We're moving ahead with putting a laboratory in Stellenbosch, South Africa, and with our collaborating hospital in London at the London Clinic. My old partner and collaborator Toshio Miyata at Tohoku University has endorsement from their campus leadership to put a laboratory in Sendai this summer, and I'm going over to help them there. So you can see we're going to have a network of laboratories with a similar approach to aging, and then we can start asking questions about what happens. What kind of intervention can we do that will either slow or bend the curve of biological aging? Is it a drug? Is it a lifestyle intervention? Is it exercise? Is it a stress reduction program? What it might be. I'm open-minded as to what might work, but I think we got a long way to go to where before we have the evidence to actually make recommendations to people that they can do anything to slow down their biological aging other than diet and exercise. Uh, but I think we all want to find that magic bullet. We want to find that drug. We want to find that intervention that is safe and effective and scalable and affordable, and find something that works for everybody in the world, no matter where you live, what your ethnic group is, what your climate is, what your culture is, and to be a part of a program like this and in this very exciting and unbelievable time in human history is incredibly inspiring. I want to make another point about our program that I think really differentiates what we're doing, and that's the inclusion of experts from all different areas of medicine and science. This is our steering committee for our human longevity laboratory, but you can see we have experts on renal function and aging, the lung, um, immunology, proteomics, nanoscience, immunology. All these different experts are included in our space and in our program. And I think these kind of programs can only be assembled at academic medical centers like ours. And that's why we're enlisting and recruiting other sites around the globe. We'd like to have some other sites around the United States. Maybe somebody who's listening today that would like to join our network and be a part of these exciting studies. But I think we can actually make a difference in the world in the relatively near future. And I think what one of the advantages we have today is I'm pretty sure that we'll be able to test interventions. Let's say it's a GLP-1 agonist. I think we'll be able to see a readout and an impact on biological age that won't take several years. I think we'll be able to see it in a matter of weeks to months. And I think if we can do that with a variety of different hypothetical uh, interventions that might impact upon aging, I think we can provide proof of concept for things to go forward on a larger scale. So, we're looking for the fountain of youth. I don't know that we have an actual sighting of it now or know where it is, but I'm really thrilled and confident actually that we have the tools to be able to figure out how to slow down aging, actually how to extend the health span of our species, and I think we're on the verge of being able to do something real in this space in the not too distant future. That kind of wraps up my formal comments today, but I'm glad to answer questions.
Thanks, Doug. That was great. I didn't want to interrupt because the story is your story and your storytelling are so great that I didn't want to interrupt and mess that up. My question list in my mind, it's exploding with questions, and I wish I had like five mics and we could all ask you five different questions to five different Dougs at the same moment because but- so what I'd like to do is jump into the PAI-1 story, then we can go through biological
Yeah,
aid, and then but before going there. I noticed that you don't have a metabolomics expert and you're not measuring metabolomics at the Potosnak. Am I saying it right? The
yeah,
Potosnak?
yeah, you got it right.
Yeah. So I
That's
don't
good. Congratulations. It's hard to say.
know.
The C is silent, PostNet.
Yeah.
You know, we're thinking about it. We're talking about it. I don't have a I don't know what the best clock is from metabolomics yet. It's expensive to do. It's got too much wobble in it. I wouldn't be surprised to have that and to include that in the not too distant future. Same, we could say the same thing about the microbiome. Same, too much wobble, and we're collecting so much information right now we can't even handle all the data. So, we're trying to be comprehensive. I think you would give us credit for being pretty thorough in terms of what we do and incorporating the functional and physiological measures as well. Everything from your body composition, using a DEXA scan to the other things that we do when you come through the laboratory. We even test hearing and sense of smell, cognitive function. But we'll get better. And part of our goal, part of our mantra is that we want to stay evergreen and we want to update that and revise it and improve it on a regular basis.
Yeah. So just to keep pushing on the metabolomics side of it, and I get what you're saying about there's so much data, it's hard
Yeah,
to know what to do with it. So I don't know if you know, but if you look me up on PubMed, I basically built my scientific career on metabolomic studies in older adults, and
yeah,
currently a part of Paolo Sebastiani's group at Tufts Medical Center,
yeah.
where we're publishing studies on the Long Life Family Study and New England Centenarian Study. So if you need a guy, I might know a guy.
I'd love to have a guy.
Yeah,
That'd be
but
great. Let's
now,
collaborate.
with that said, I would, even though Paola and Stefano, I don't know if you know Stefano Monti, even though they've developed metabolomic-based clocks in, centenarians and their offspring,
Yeah,
I'm not so hot on that trail. What what uh what interests me about metabolomics is that most of these, and now granted, Jessica Laxey, I don't know if I'm saying her right. She's at Harvard. She recently published the Omic Age, which is also
yeah,
offered by True Diagnostic, and they do include some metabolites, but they're epigenetic approximations of the
yeah,
metabolites.
yeah.
So that's another topic, a rabbit hole that I'd like to go down before, but before
Mhm.
going there, I think most of the current biological age clocks, whether it's PhenoAge, Morgan Levine's blood-based biomarker clock, which I think is great, and
Yeah,
I don't just say that it's great, but all the published literature on study after study that's used it in various outcomes. But it's got weak spots. It doesn't include CVD risk, and most people are going to die from CVD risk. So
yeah,
you've also
yeah,
got to measure that on the side. So the long story short there is with the metabolomics, there are things that aren't covered, even in the epigenetics,
yeah,
and without going through the list: kynurenine to tryptophan ratio, the androgens, which decline during aging,
yeah.
bile acids, primary and secondary. There's a rationale for the depth of data would be additive onto the clocks you already have. So it sounds like I'm selling myself to the
No,
post.
I agree with you know what you're preaching to the choir. I hear you, and there are sort of logistical limitations of what we can do. We when we bring somebody through the laboratory, it takes about 2.5 hours to go through all the testing stations. Okay, that's not too bad at all. But then there's a cost related to doing the molecular profiling in particular. And at this point in time, we don't have unlimited monies, but I think we've got enough preliminary data. We've demonstrated that we can actually do this and do our measurements in people. Now we're trying to we're putting some grants into the NIH and the NIA and the NICCH to see if we can't get support to do these studies on a larger scale and to support the the molecular profiling that we need to do, including metabolomics.
So it could be something, a cost that could be absorbed by the patient. I mean, so we send our samples, or Paula's group has sent their samples out to
Yeah,
Metabolon. And
yeah.
then if the patient picked up that cost, and then the POTUSNAC, doing the analysis, even though Metabolon does some analysis, but it's very, very
It's a
super.
little, it's a little bit of a tightrope about charging people to do research. We don't charge it. If anybody goes through our program, we don't charge them a penny. Okay,
Interesting.
this is research, so we support it and we do it uh, as part of our mission. We do have what we call the Human Longevity Clinic. It exists in parallel with us, and that's a fee for service program, and they are able to do, to charge for that. But unfortunately, in 2026, insurance doesn't cover this yet. I
Yep.
think hopefully it will someday.
Yeah. So I'm sure that your group and others will find ways to quote unquote lower biological age in association with improved outcomes, but that's mostly going to be probably cross-sectionally. The
Yeah.
challenge, and this is one of the reasons I left academia, is that I don't see a pathway at the NIH or even in independent private funding for N of one studies. And I think this is actually where the golden ticket is for quote unquote longevity. It's testing yourself not once a year, not twice a year, but literally as many times as you physically can. And for a various large group of biomarkers, both from the physiological, as you've got, the cognitive function, physical function, gait, retina age, all of that, but down to the molecular, as you've got, the proteomics, hopefully metabolomics at some point,
Yeah, yeah.
even putting in epigenetic age there, but I can, there too, I don't want to go into that yet, but so it's tracking that over time. So all right, what I'm trying to say is people taking GLP-1, they'll lose weight, they'll probably improve biological age, but then they're still going to have some aging rate after that. And if they just stop measuring, they're not really seeing if there are ways they can slow their aging rate, flatten that mortality curve by slowing their aging rate. Whereas the N of one studies for people like me who are testing 8 times per year, I'm blood testing 8 times per year,
Yeah, yeah.
and then I'm taking the average of that year to year and looking for correlations in my diet to see what I can do. What is the recipe for flattening, not just biological age, but all these other biomarkers that aren't included? So are there plans to, I mean, maybe this is the longevity clinic that the POTUSNAC would do, but um, tracking longitudinally rather than cross-sectionally. What are your thoughts?
Um, I think what you're talking about is valuable. I think we want to find some interventions that are that we can confidently state can be distributed to more than one person. We need some p-values. That means we got to enroll some people and test some interventions. And that's sort of where we're at. I think that in particular, since we have this mission to figure out how to slow down aging in people that are disadvantaged for one reason or another, that gives us very selected populations, people with chronic HIV, um, you know, adult survivors of childhood cancer, people that have had toxic exposures in their life, people with chronic inflammatory conditions. There's so many people like that. I could take you and walk you through the floors of Northwestern right now, and I could show you all kinds of people that are disadvantaged with regard to aging. So our mission, uh, it's part of our mantra is to do something for people that are disadvantaged in that space. Other places can work on figuring out how to take healthy people and have them live longer. And hopefully, what we'll find will be applicable to anybody and everybody. We want to find interventions that are scalable to the 99% of us in the world.
Yeah, the challenge though, so I mean, there it would be eat less calories and exercise more often, as that's going
Yeah,
to move the needle on most biomarkers. But
yeah,
the challenge there is which goes back to this idea of precision nutrition, where people
yeah,
not just having to track the gross physiologic biomarkers, but also the inputs, diet, exercise,
yeah,
wearables, sleep,
yeah,
all of this stuff, and then
yeah,
figuring out the recipe. But even there too, it's a challenge. For example, when considering CVD risk is it's in my family tree, so that's probably what would kill me.
yeah,
And for most people too, if I eat a higher fat diet, I'm not talking about a ketogenic, carnivore, no carbohydrates. My fiber intake is 70 to 75 grams per day right now. I'm on a
yeah.
very high fiber, mostly vegetarian diet, but
Mhm.
only because the biomarkers say so. So if I go beyond a certain limit for fat intake, total fat intake, and this isn't from processed foods, this is, whole food based, nuts and seeds, no oils, just whole food
Yeah.
based.
Yeah.
eh, I'll optimize my triglycerides will come down real low. My HDL will go up into the 60, 60 range, which is close to optimal. My
Yeah.
protein A will be lower than if I'm on a high carb diet. But where I'm trying to go with that is that diet may optimize CVD risk. But when I look at things like DHEA sulfate, which is a
Yeah.
neurosteroid and declines during aging, I've got like a 50% decrease over the past year, which can't
Mhm.
just be from aging.
Mhm.
So the balance is I see precision nutrition and precision therapeutics, whether it's metformin or berberine or whatever it may be off the list, is looking at as many of these biomarkers as possible. And then it's got to be a little bit of give and take. Well, I can reduce CVD risk, but if I do that, my androgen levels may not be as high as they should be, right?
Yeah.
So,
Yeah. Yeah. Well, um, we're going to be try to be as nimble and as adaptive as we can be in terms of testing interventions that have the potential to do something. We hope that we can scale this up and have enough of a network around the globe that we can actually get some numbers enrolled and start asking questions like, specific dietary interventions. Or, and that was a pretty interesting story that came out that was, I think, highlighted on NPR last week about the effect of music on some measures of biological age. The spectrum of interventions is mind-boggling, right? I sort of want to do a study comparing a prospective study comparing Mozart to Metallica, and what's that do to your biological age?
it made it.
Here's a fun one I want to test. This little blister pack. These tablets are PAI-1 inhibitors
Hmm.
developed in Japan. They are in late phase human testing. They're the company that developed these are really thinking that PAI-1 inhibitors might be an important treatment in a variety of different cancers related to that issue I talked about earlier today. But we've just applied to the FDA to get an IND to test this in our longevity laboratory. Hopefully, we'll get the green light to start testing this and seeing what it does to on biological age and non-Amish individuals in and around Chicago. We'll see.
All right, so that's a perfect segue. Okay, I have a quick recommendation if you're interested in an intervention that may make a broad impact
Yeah,
dietary.
yeah.
Would be mushroom intake. So,
Ooh,
so mushrooms contain lovastatin. Most people don't know the mushrooms contain a statin.
mhm.
So you could get the CBD improving, improved cardiometabolic health. They contain ergothioneine, which extends lifespan in flies, yeast, and worms. They
Yep,
contain spermidine, which extends lifespan in mice.
yep.
So, but the question is dose. Do you have to go to 200 grams per day or 400 grams per day? It's probably not going to be 30 grams per day if anybody's even eating it besides me, in high amounts at all. So all right, that would be my best bet. But all right, so going back to the PAI-1 story, which I had a great segue there.
Yeah,
So, and I'm sure I think, in the era of AI, people are going to think that I've picked this off of AI, but I promise you I haven't. So in terms of the why PAI-1 increases during aging, right?
yeah.
I get that it does increase during aging, but in terms of the why, it's a serine protease inhibitor, which means that when serine proteases are released, whether it's by neutrophils or by what's the other cell type that
Well, we have
uh,
a bit, the serine proteases that PAI-1 is designed to inhibit, like TPA and UPA, they mostly come from your endothelial cells in your vasculature.
I get that, right? That's a very direct story. I don't dispute that at all.
Yeah,
The endothelium.
yeah,
So I knew where I was going with this. So elastase is a serine protease that's produced
yeah,
by neutrophils and that's produced by the pancreas. So
yeah,
neutrophils increase during aging. Systemic microbial burden increases during aging. Older
yeah,
adults rate is
yeah,
yeah. So I
yeah.
wonder if the age-related increase for PAI-1 is somewhat in response to an increased systemic microbial burden. What are your thoughts?
I think there are a lot of things that probably contribute to the increase in PAI-1 that we see as a function of age. Some of it's physiological, some of it's related to BMI. and the visceral contribution to circulating PAI-1 levels. PAI-1 is a marker of senescence. As cells adopt the senescent pathway, they make more PAI-1. And that there's sort of an amplification loop there. And PAI-1 drives senescence alone, let alone being a marker of senescence. So that could be part of it. There are other factors that likely impact upon um, PAI-1 production or its half-life in the blood. Uh, it's one of the key things that determines how much PAI-1 you have in your blood is your systemic inflammatory state. So PAI-1 is an acute phase reactant. If you are inflamed, you will have a high PAI-1 level. So I think that kind of relates to what you're asking there.
So I was going to ask you about that. I'm glad you mentioned that it's an acute phase reactant because there's conflicting data in the literature, at least in rats, where from a long time ago, studies that were done a long time ago that were saying that it's not, but that it is an acute phase reactant. That would tie into this story that maybe during aging, with the increase in systemic microbial burden, maybe neutrophils get to the point where we need the extra boost with PI1
Yeah.
to for whatever reason, maybe there are bacterial serine proteases that negatively affect cellular function. I don't know if anybody's even studied it. The
Yeah.
other the other thing I think is interesting is that so elastase, which is also a serine protease, that's produced by the pancreas. So every
Yeah.
time we eat, we get that released into the small intestine. And then the thing is, though, that's not a perfect system. It shouldn't be in the blood. Elastase
Yeah.
shouldn't be in the blood
Yeah.
unless there's an infection. But I don't know if you're familiar with the paper Aging by Autodigestion by Geert Schmidt.
Oh yeah. Yeah. Yeah.
Yeah, so they saw that elastase levels were increased in heart in old aged I think it was mice.
Hmm.
So within that context, couldn't one make the argument that the release of PAI-1 during aging, and when considering elastase may be higher in the blood, whether it's because of neutrophils or it's increased small intestinal leak, could that increase in PAI-1 be to help, it's basically a marker of small intestinal leak for enzymes that shouldn't be in the blood.
It's an interesting hypothesis. I'm going to have to think about that a little bit. The the you know the story about where PAI-1 comes from and its synthetic sites, I think, is a little bit complicated, and I think it's I think it requires some um, thoughtful reconsideration. For a long time, um, the community that work on this sort of the idea has been that the PAI-1 that circulates in your blood, well, it's made in your liver, and it's made in your endothelial cells, and it's made in fat. Well, the paper that we published in the JCI in December looked in aging mice, in a mouse model of vascular age, and we found that the PAI-1 that came from the blood vessel wall was from the smooth muscle cells, not from endothelial cells. We did single cell RNA-seq, and I think that's a surprise. I think you have to have diseased endothelial cells to make PAI-1. I think we've also learned that we've we've done things like shut down hepatic PAI-1 production with an siRNA, and we see that has systemic effects on aging and metabolism independent of visceral fat. And that might be misguided too. So it makes me think that the liver PAI-1 might be a more important contributor to the aging related changes that we see systemically more than we might have imagined till we had the tools that we have today to be able to precisely identify where it's coming from.
Yeah, it's tough to separate it out though, because as you mentioned, visceral fat produces it. So you've got hyperinsulinemia. So you've got visceral fat, hyperinsulinemia, more PI1.
Yeah.
Okay. If the insulinemic state, probably also pro-inflammatory, more CRP, that's
Yeah.
going to
Yeah.
trigger the endothelial cells to produce PI1. But
Yeah.
then where it gets super interesting for me is that systemic microbial burden story again, which if it is indeed, as you mentioned, an acute phase reactant, that
Yeah.
highlights the liver and producing it as some kind of help or additive effect on the immune system to counteract something that microbes may be doing. Um,
Well, there's no doubt that inflammatory cytokines stimulate PAI-1 production. One of the most powerful drivers of it is TNF alpha, but interleukin 6 works as well. The other molecular drivers or regulators of PAI-1 production include the circadian clock mechanisms. PAI-1 in your blood peaks in the morning, goes down late in the afternoon. We have the circadian rhythm every day, and it's incredibly powerful in driving PAI-1.
so I've got it. That's another rabbit hole I've got to go down. I didn't even consider the circadian effects, which also change during aging. So that's
Oh,
great
yeah.
insight.
Yeah. Yeah.
So in terms of, is it possible? And I was actually on my list, TNF alpha, the SASP proteins,
Mm.
how they relate to PI1. But is it possible for someone who's relatively lean and exercises and fit, who's probably has low insulin, low visceral fat, probably low, not super low fibrinogen, but youthful levels of fibrinogen with low HSCRP. Within that context, can one still have high PI1?
I think you can have. I think that you can have relatively high PAI-1 even in that circumstance. We did a very interesting experiment a few years ago, and it's in mice, but follow me. So we wanted to ask the question based on what we saw in the human population in Indiana. We want to ask what does lowering PAI-1 by half, taking in a heterozygous mouse, what does that do to hepatic gene expression? And so we took an open mind to it, unbiased analysis, RNA-seq of the liver, wild type mice versus heterozygous mice. And the fellow working on the project, came running in my lab when the results came out. He goes, you know, guess what gene is most upregulated when PAI-1 is knocked down by half? I said, I don't know. He goes, FGF21. FGF21. Very important in metabolism, glucose metabolism, and maybe an anti-aging peptide. What's what's the gene that went down most when we lowered PAI-1 by 50%? I don't know. He, PCSK9. PCSK9. I said, you can't make this up. Two of the most interesting targets in modern medicine are regulated by PAI-1 at a level where, a normal mouse, you can't measure PAI-1 in their blood. And we lowered that by 50%. And that had enough effect on the liver to register things like that. And we found that was the same. We could see the same effects by feeding mice the PAI-1 inhibitor that Professor Miata has delivered. We've seen it in mice when we've with the reverse engineered mice that have the genetic variant that the Amish have. So I think there's a story there. You don't want to have zero, but I think anything is almost too much as you approximate having none.
Could you then call it maybe an immunometabolic protein, not just a clot dissolution, but FGF1 metabolic in well, actually metabolic would be the cholesterol story too, the PS.
Oh yeah.
Yeah,
And triglyceride, it's involved in triglyceride assembly. And so I think it's a cardiometabolic
yeah.
peptide, but immuno is I think I'm with you. I think that's part of the story I was trying to tell that wow, it's involved in metabolism and uh, salt excretion, phosphate excretion, brain, neuroplasticity, all these kinds of things. Yeah.
So PI1's been on my radar for a while, but the challenge is that U shape, how
Yeah.
low, too low. So when consider you've studied it for 40 years, what would be like an optimal target for PI1 youthful levels?
That's a good question. The average level that we measure in adult human beings, we've measured it in thousands of people now over the years, is probably about something less than 20 nanograms per mil for the antigen. So something less than that, maybe 10, I think maybe 10 might be an optimal target. Uh, we see a lot of people with too much PI-1 now, and mostly related to their weight. Uh, so I would encourage, I would work on that group first, trying to get it normalized, get it under 20. Uh, and I don't think there's any way you can make it zero in anybody with anything that you do. So I don't think you're going to be at risk for any of the long-term consequences of having no PAI-1, and they're real. And if somebody does come up with drugs that can block it or reduce its production, they're going to have to be mindful of the fact that you can't titrate it too low. It's not good. Uh, it's not life-threatening. I mean, the people that have homozygous PAI-1 deficiency, they live to be adults. They're not, and but their lives are a little bit complicated. So you don't want to have zero.
Yeah, I wish you had shown the data on the heterozygous Amish because that lifespan curve is like it's like a almost a 10-year increase in median lifespan for
Yeah.
people who don't know, which was another reason why I was like, all right, I've got to get you on. This is great data. Couldn't resist the PI1 story anymore. But the argument against PI1 in my mind for a long time was even though it's a strong contributor, the epigenetic approximation of it, and I think that's an important thing to highlight. Even though a lot of that story matches what you see in the human studies,
Mhm.
like do you know the correlation for the DNA M versus the actual plasma or serum level of PI1? Like, was the correlation 0.2, 0.8? Was it strongly correlated? Greater than
Oh,
0.7?
yeah, that's how they selected the CPG islands that derived it. It's highly correlated with plasma PI-1.
But something could be highly correlated statistically, but it may
Yeah.
not, but maybe not in terms of the correlation coefficient, which the reason I raise that is when considering so what I'm trying to get at here is when you look at grim age, which I
Yeah.
have no disagreement with, it is literally it's study after study. It's the gold standard clock for mortality risk. I wouldn't necessarily say biological age, but mortality risk,
Yeah. Mhm.
risk of death for all causes. But in that study that you cited, they put the data for the original 7 biomarkers. Grim age version 2 now has HSCRP and HbA1c.
Yep. Yep.
But the 7 protein-based contributors that are in Grim age, they looked at the correlation for each of those proteins and how it predicts the overall Grim age score. And things like cystatin C, GDF15, B2M, the
Yep.
correlation was like 0.8 to zero point 0.9, but for PI1, it was only 0.44.
Mm.
But the
Mm.
reason I go through the story is not to suggest that PI1 is less important than those other biomarkers.
Yeah. Yeah.
It's this DNA M approximation of PI1 that I wonder what else it's picking up to have such a strong mortality risk association in the epigenetics. I don't think it's just picking up PI1. It's got to be picking up other stuff too. So I don't know. What are your thoughts on?
Yeah. I think that I think it probably represents the multiple targets related to PI-1 that are also influenced, whether it's glucose metabolism or other factors that are in the field of peptides and processes that are regulated by PI-1 itself. I think that's sort of my explanation, but you might be right. I don't know. Good question.
If you had to pick, which would you rather have, the plasma or serum measurement of PI1 or the epigenetic approximation?
Well, the epigenetic approximation is much easier to do, I think, overall. The problem is that, and this is been part of the challenge being somebody that works in the field of PI-1, is that a given individual has a circadian variation. You've got to measure things the same time of day, or you get nonsense. You get the variability becomes uninterpretable. So that's a problem. You can't measure it in serum. You can only measure it in plasma. If in serum you get platelets dumping, and they are a big storehouse of PI-1, so that confounds the measurement as well. So it requires some precision, handling blood specimens carefully, spinning them down, putting them in the freezer, and doing it at the same time of day. All that is logistically challenging. It's easy to take a specimen of DNA and put it on a piece of filter paper and do Grimage. I mean, it's much less technically challenging.
Yeah, just to push back a little bit on that. So I've
Yeah,
had HSCRP measured. I won't mention the company. It's a part of
yeah,
their, yeah. And so on the same day, I'm also measuring it in blood. So I have the DNA M and I've got the actual biomarker, and they're significantly different. Like
yeah,
their
yeah.
epigenetic approximation is actually high, quote unquote high, but yet I've had HSCRP that's less than the detection limit, 0.3 milligrams per liter, something
Wow.
30 tests.
Yeah.
Makes me, so if it's me, so I get you on the PAI-1, and it's because of your work that I'm going to measure it for the first time for my June test. But now I'm worried that, we're considering, you're saying that there are technical limitations. Granted, I measure at the same time for every test, so that'll be
Yeah.
a
Yeah.
minimal
Yeah.
factor, but
Yep.
hopefully
Give me a call after you have it done and we'll talk about
that sounds good.
it.
All right, so are there current and future projects that you're working on that you're excited about now, besides biological age, or in addition to biological age, PAI-1 and other stuff?
Oh yeah. Our focus is pretty clear, and we wouldn't be having this conversation today if I had not focused on PI1 for, the last few decades of my life. So that continues to be a key component of what we're doing. I want to help bring the PI1 inhibitors into the clinical testing arena and see what they can do. But our longevity lab is um, is already in the process of starting to do prospective clinical trials to see if we and different patient populations to see if we can bend the curve of aging. Our first study is in relatively healthy middle-aged individuals, and we're putting them through an online stress reduction program developed by my colleague Judy Moskowitz here. She's one of our psychologists. She has an online tool that tens of thousands of people have used around the country, and a series of exercises you do over several weeks. It reduces your stress level and improves your mood. And we all know that stress makes us age, right? Well, does de-stressing you actually do something to your clock in one way or another? So we are our first pilot study involves enrolling 80 patients and following them for 6 months. We'll finish enrollment next week. So that's our first one that we'll kick off. We got another one that's going to start this summer. We're going to test a novel dietary supplement consisting of a sugar that's only found in breast milk. But if an adult eats that, it actually resets your microbiome to a more juvenile state because,
Galacto-oligosaccharides?
yep, it's a fucose. We're going to test and see if that does something systemically to biological age and population of patients that we know have accelerated aging. We got all kinds of collaborators other collaborations that are going on. We're doing a selected study of adult survivors of medulloblastomas, an unusual childhood brain tumor. They go through a terrifically challenging treatment program to survive that. What's that do to their biological age? What do they look like when they're 20? And and that's gonna, depending on what works out, that'll be representative of the sort of population where we want to test high probability drugs to see if they can bend aging, like SGLT2 inhibitors or GLP-1 agonists. Can you do something for people that have had to go through something like that in life? And got several different populations of patients that are undergoing treatment for cancer or survived cancer treatment to see what that works like. My friend and collaborator Lorenzo Gallone over at UIC in town has set up a laboratory that we've helped him get up and running. He's part of our steering committee, but he's a transplant nephrologist. He's testing the Putin hypothesis over there. If you get a transplant, an organ transplant, does that change your biological age? And does it make a difference on if the donor is young or old? Amazing hypothesis. And they've already enrolled quite a few patients already. And so I think that's going to be very, very interesting and fascinating to think about. But, the world is kind of our oyster now. There are so many questions and so many opportunities. And the fact that we have a lab and we have the tools to be able to measure biological age in a meaningful way makes me think that we can actually make some progress in starting to solve this problem.
Definitely. Well, I think you're great, Doug. I think what the work you do is great, and I'm looking forward to hosting your work again at some point in the future on the channel.
Thank you so much. Enjoyed talking to you.
Have a good day. Bye.
Bye.